Welcome. My name is Paula Hemdel, and I'm a freelance trainer with the European Center for Clinical Research Training, or ECCRT. I'm pleased to moderate this free webinar on adherence-informed clinical trials to optimize drug development and enable precision medicine that is being presented by Dr. Professor Bernard Vreyens. This is a very interesting topic. It's related to the adherence of study medication in clinical trials and how it can affect the outcome or the quality of the trial. And Professor Vreyens is the Chief Executive Officer of the Advanced Analytical Research on Drug Exposure, or the ARDEX group, and a professor of biostatistics at Liège University. He's a co-author of 7 books, over 100 peer-reviewed scientific papers, and also inventor on 6 patents. He's very much into the patient adherence, compliance, and persistence, and is very active in several EU and US collaborative projects around the theme of adherence to medication. So I'm really pleased to go ahead and pass the microphone over to Dr. Vreyens to talk about this interesting topic. Thank you.
Thank you very much for the introduction. Hello, everybody. It's a pleasure to talk about this topic, medication adherence. And my objective today is to convince you that medication adherence is a major source of variability in drug response, from drug development, prescribing, dispensing, pharmacokinetics, pharmacodynamics, and ultimately drug response. I will focus during this presentation a lot on the need to integrate adherence and to have adherence-informed clinical trials to address the problem of medication adherence in clinical trials. And this is a reference to an editorial that we wrote a few months ago in Nature's Reviews, clearly calling for adherence-informed clinical trials. But also at the end, I will focus a little bit on initiatives like the Precision Medicine Initiative in the US or similar initiatives in Europe, trying to identify through gene some variability in pharmacokinetics, pharmacodynamics. And I would like to stress that this is only possible if medication adherence is taken into the picture. So to address appropriately the problem of medication adherence, we need sound measures. And I have classified on this slide the different measures for medication adherence as being biased versus reliable, or as being rich sampling versus sparse sampling. And for example, when we typically do pill count in clinical trials, we have a measure that is extremely biased, but also a measure that is sparse. And I will show you some examples. Very often when we are concerned about medication adherence, we do therapeutic drug monitoring, measuring blood concentration, which is a reliable measure, but an extremely sparse measure because it gives you an idea of the adherence at the time of sampling, but nothing in between clinical visits. Patient diaries have been extensively used. They can deliver a rich sampling, but typically they're biased because paper diaries are typically filled in the waiting room before showing up to the investigator. And electronic diaries are often combined with reminders, where it's very easy to say, yes, I take, while it's completely disconnected from the medication intake. What we have developed at ARDEX for many years, for now 35 years, is the idea to put a chip in any type of packages. So the idea is that you put a chip in any type of packages so that you record in real time when the patients access the medication. It's a measure of pill in hand that we have called medication event monitoring system. And most of my presentation will really be focused on the data collected through MEMS systems. This is an example of the type of data that we are collecting. So here on the x-axis, you have the days of follow-up in the clinical study. On the y-axis, you have the time of drug intake. And every time there is a blue dot, it's a dose taken. So here you can see a patient taking a twice daily dosing regimen. he takes the morning dose about 7 o'clock in the morning and takes the evening dose about 7 p.m. in the evening. You can see the patient was once 2 minutes late here to take a medication, but this patient is almost perfect, and there is exactly 12 hours between the morning and the evening dose. So this is a real patient from our database, but you can imagine that many patients will deviate from this perfection. A typical deviation that we see in many clinical trials is typically this more variability in time of drug intake. So this patient, for example, takes the medication early in the morning on weekdays, wakes up later on weekends, takes the medication late in the weekend, and you can also see that in the evening there is more time variability in the time of drug intake. But still, this is a very good patient as he never missed a single dose over the course of the study. And then you have this type of patient where you see the morning dose is pretty well taken at the beginning, the evening dose is already problematic, and then you see as we move on in the study, the dosing, the adherence to the medication is decreasing and really worsening a lot, especially in the evening, and then there is a full treatment discontinuation before the scheduled end of the study. Another example is typically a patient here who has a lot of trouble at treatment initiation, and then he is doing much better, and about halfway of the study he is stopping treatment definitively, but still showing up at the last visit and didn't report that he was stopping the medication. So just to give you an idea, here I have taken 6 patients because very often we consider that 80% adherent is a good adherent. And you can see here 6 patients, and all 6 patients have taken exactly the same amount of treatment. They have taken 81% of their prescribed doses. The 3 patients on the top lines are once daily dosing. You can see random missed doses. You can see taking the dose almost perfectly, but then a little holiday and then a bigger holiday. The last patient on the top line, you can see how often he changes behavior, going from wide variability, missing a few doses, stopping, starting very well, missing more, a few doses, more doses, and so on. And then at the bottom, you can see a patient a twice daily dosing regimen where the number of missed doses are increasing over time. A patient having problem at the beginning, but then almost perfect afterwards. And a patient having clearly more problem in the evening than in the morning. So you can imagine that those different behavior will lead to different clinical consequences, but also if you want to build an intervention and you know the behavior of the patient, it's much easier to have a focused discussion with the patient to manage his adherence. So those figures show you also that we have different behavior and that we want to understand those behavior. And that's why it's important to use the ABC taxonomy when we talk and we analyze medication adherence. So we have defined the medication adherence as the process by which patients take their medication as prescribed, but we have to recognize that this is a dynamic process over time. It can change over time. And there are 3 key elements in medication adherence. Once there is a prescription or a treatment initiation in a clinical study, first of all, the patient has to initiate therapy. Once the treatment is initiated, the patient has to implement the dosing regimen. That means taking it once a day, twice a day, with food, without food. And when we think about polymedication, this implementation can be pretty complex. And finally, the patient has to persist with treatment for a long period of time, especially in chronic diseases where it may be lifelong, or in clinical trials where we have a defined duration of follow-up for each patient. And what can go wrong? Either the patient does not initiate, it's a dichotomic outcome. Either the patient delays, omits, takes extra dose, it's a dosing history. Either the patient discontinues treatment too early, and that's a time to event. So statistically speaking, those 3 elements are very different in nature, and we need to study them separately. So I will review and give you one example for each in the next 3 slides. First of all, in treatment initiation in medical practice, we know that 20 to 30% of the patients do not initiate a new treatment that is prescribed. And when we think about diseases like hypertension, hyperlipidemia, diabetes, we see that we are close to 30% of adults who are not initiating a treatment prescribed for a chronic disease. So this is a huge problem in medical practice. It is less a problem in clinical trials, but initiation is the beginning of medication adherence. Then, to illustrate what we mean by implementation of a dosing regimen, this is an average patient, I would say, in a clinical trial. So here you have the days of follow-up on the x-axis, you have the time of drug intake on the y-axis. This patient is followed for 632 days. Every blue dot is a dose taken, every red line is a missed dose. So there are 14 days with double dosing and 115 days, that's 18%, with no dose at all, which makes this patient an 84% adherer. Typically, that's how we call it. But when we think about medication implementation, we have always to put it in line with which treatment is this patient taking. So for example, I can say this patient was taking a cardioaspirin between 2011 and 2012. That means that because there are no consecutive missed doses there, almost no major consecutive missed doses, with a cardioaspirin that has a duration of action for 2-3 days, probably that this patient was very well covered. If this patient was prescribed a once daily anticoagulant, for example, this patient may be at high risk with an 84% adherent. So how much implementation depends really on the drug's forgiveness that the patient is on. Finally, we know that about 40% of the patients discontinue treatment after 1 year. And here I have a summary of 17,000 patients almost coming from 95 drug development clinical trials. So we are talking here about sound, well-designed clinical trials in which we have used the monitor, the MEMS monitor. And you can see that first of all, you have the blue curve. So the blue curve gives you the persistence to the treatment over time. And you can see that after 1 year, about 40% of the patients have discontinued treatment. And even on day 1, you have a 3% non-initiation. You see the blue curve dropping by 3%. So that means that even on day 1, in well-designed clinical trials where the patients are highly selected to participate, about 3% do not initiate and about 40% stop within 1 year. The red curves gives you the proportion of patients who take the medication as prescribed over time. So for example, if I take day 100, I have 80% persistence, but only 65% implementation, adherence. That means there is a gap here of about 15%. So that means that every day, every consecutive day in the trials, among the patients who are engaged with the therapy, about 15% of them don't take the medication as prescribed. And those are the 15% I mentioned in the previous slide. So we have also looked at persistence across therapeutic areas, and you can see that we have the worst persistence in depression. It's in depressive patients that we see the worst persistence, and we have to think about all diseases that are related to depression, like multiple sclerosis, cancer, RA. Those are diseases where the level of depression is high and where it is a problem for medication adherence. So that allows me to define the adherence gap. Very often, and I'm sure we are familiar with this, we say that in drug development, phase 1, phase 2, phase 3, we have a true estimate of efficacy because everything is perfect in trials. Once the drug is going on the market, we have an estimate of effectiveness. And that's really what the payers are looking for, is how is the drug working in medical practice. And we know that we have a gap between efficacy and effectiveness, and it's always difficult to justify that gap. But based on the data I just showed you, I think it's worse. In reality, we have non-adherence in the clinical trials as well, especially as we move away from phase 1, in phase 2, phase 3, when we are working with ambulatory patients, we have suboptimal adherence, and we still do better patient selection and patient follow-up because we are in a clinical trial. So that means that the payers are looking for an estimate of effectiveness, the regulators are looking for an estimate of efficacy, and we provide them through the clinical trial process an estimate which is in between and doesn't answer any of the questions. And that has major consequences. First of all, there is a high risk of failure because of lack of effectiveness. We have a poor estimation of toxicity, and very often, to compensate for the lack of effect or of efficacy due to medication adherence, there is a tendency to go for treatment at doses that are higher than needed, and we end up with inappropriate dosing regimen in the marketplace. And those are real problems that should be better addressed in the future by using adherence-informed clinical trials. And this is becoming a regulatory priority at the FDA, but also at the EMA. I wanted to show you one example of this adherence gap, and it's a seminal example in the PrEP. So it's using antivirals to avoid contamination of HIV in a high-risk population. And I just review this very briefly, but the first study that was done and that was performed had an efficacy of 6%, which was not different from zero. And then when they looked at the level of adherence through blood concentrations in this study, they realized that only 1/3 of the samples contained the drug. So there was a very high level of non-adherence in this clinical study. And then the teams designed adherence intervention to improve adherence. And you can see as they were able to improve adherence, they were improving effectiveness of the treatment. The last study was a study that used electronic monitoring of medication adherence. And at every month, at the clinical visit, the data would be downloaded and discussed with the patients. And this delivered about a 97% adherence with 100% efficacy. So here you can see that the clinical study effectiveness ranged from 0 to 100 with an average at 47, which was a real challenge for the ITT analysis and the FDA to put this drug on the market. But finally, they accepted, they put the drug on the market, they said it only works if it works, it only works if you take it. And those were the conclusions from the FDA. Adherence based on self-report and pill count was not reliable because in those studies, the pill counts were quite high. The reliability of adherence data should be improved. No covariate can have a larger impact than not taking the drug. And adherence could provide valuable information for efficacy and safety, and more attention should be paid in regulatory review. Just one slide to show you a striking example, because we think that when we are involved in life-threatening diseases that adherence is perfect. But this is a study we did with Alabama University, and we did monitor about 600 children with acute leukemia, and 40% of those children didn't reach a level of adherence that was acceptable, that led to an increase in relapse rate of almost threefold increase in relapse rate. So even with children in oncology, adherence is not perfect. And very often, and that's what was shown also in the study, is that when adherence is suboptimal in practice and is not measured, then the treatment fails. When the treatment fails, the disease progresses, or you have acute events sometimes. And what we typically do, we give more complex treatment. And the more complex is the treatment, the worse is the adherence. And this is a very costly loop in medical practice that requires also adherence-informed prescription. So we need adherence-informed clinical trials, and we need adherence-informed prescriptions. Because today, most of those dosing errors go undetected. And those are examples from clinical trials we were involved in. So this is, for example, a study where we have tablet count based on electronic monitoring on the x-axis. We have pill counts, traditional pill counts based on counting return tablets. And you see the bias in pill counts in about 20% of the population. And typically, those are the worst patients who are really poor adherers in the study. They drop the pills before showing up at the visit. This is a comparison between last reported dose or last two reported dose in pharmacokinetic studies based on electronic monitoring and based on patient self-report. And you can see again here about 30% of self-reported last intake, which are completely inappropriate. leading in major trouble when trying to fit the POP-PK models. And those two last figures at the bottom, those are studies showing you the difference between persistence in brown according to electronic monitoring and persistence according to self-reported dropout by the patient. And you can see here, for example, in this first study that at the time of primary outcome evaluation, there is a 22% difference between the patients who really stopped taking the medication and the patients who reported to stop the medication. And that's critical when you analyze the data of this study. And this was a very well controlled PK study. And you see that because it's PK very well controlled, at the end, the proportion of self-reported dropout and electronically monitored dropout are very similar. But there is about a 50 days delay between the day they really stop and the day they report they stop. And those bias are really, this is really important to know in the assessment of the clinical trial. And it's becoming a regulatory priority, as I mentioned, at the FDA, at the EMA. The OECD just launched a big call for better adherence in practice. And I have also to say that multiple therapeutic area, like here, the NARC guidance that is coming for anticoagulant treatment, and there will be more in pain, oncology, dermatology. There will be disease-specific calls for better management of adherence in clinical trials. So now we are in the solving. How can we solve the problem of medication adherence? And typically when we want to solve something today, we go on Google and say, how can I solve medication adherence? And you will find about 3,600 apps about medication adherence. One of them pretty often downloaded is MediSafe, and you can see their message. They capture if you miss a dose, and if you miss, if Joe forgets, his wife receives an alert and she has to take care of it. You have Vika, they watch you out of the sky, and very similar, if you forget your medication, your wife will remind you. Seems to be a man problem based on Google, but I don't think it's a man problem, it's a worldwide problem of striking magnitude. Then some application will ask you to take a picture every time you swallow a pill and send that information to the cloud, so they are proving you are swallowing the pill. Or more recently, there is the Proteus Smart Pill. It's watching and proving that the pill is in the stomach, and it goes with a patch and an app. It's quite complex. I just wanted to warn you that we have to be we have to take a lot of cautions about those apps because it's extremely intrusive for the patient. It delivers highly biased results because the patients who are using those apps as a very subset of the population. And they are all based on reminders. And I would say time-based reminders. They say the problem is patients forget, so let's remind them at 7 o'clock in the morning. And when we question patients, only 7% of the population is time-based. 93% of the population, when they take medication, is even-based. They take it when they get up, when they take their first coffee, when they take lunch, when they go to bed. So we have to find another ways, and we have to build habit in patients differently than reminding the patients at fixed time, because it only fits 7% of the population. So what we really need is a sound management of adherence, and this is a real system approach. So the idea, the definition is the process of monitoring and supporting patients' adherence to medications by healthcare systems, patients, provider, and the healthcare has to change their model to address medication adherence. But the most important is the objective. The objective is not to make patients swallow pills. The objective is to achieve the best use by patients of appropriately prescribed medicine in order to maximize the potential for benefits and minimize the risk for harm. And on the next slide, I will illustrate what I mean by this. Because very often when we give a treatment once a day here as an example to a patient, we assume that his pharmacokinetics, for example, they will reach a steady state after a few days and they maintain a steady state over time. And this is the picture we have all in mind when we say this patient is on treatment. The reality is very different. Patient miss a dose, take an extra dose here to compensate a missed dose the day before. They take a little holiday, a bigger holiday, they miss several doses, and you see that there is much more variability in the dosing history process. which can sometimes lead to toxicity and sometimes lead to periodic loss of effectiveness and also emergence of drug resistance in some cases. And this is what we need to much better understand during the drug development process. What we have shown is that when you share this information with a patient, when you can measure a patient for a while, this is one example of a patient taking a statin, for example, you measure his behavior and then you have a focused discussion with a healthcare professional. In this case, it was a pharmacist, but it can be an investigator, typically in a clinical study. You can drastically change the behavior of the patient. And in an EU-sponsored study, we have identified about 30 clinical trials, randomized study, showing that feeding back that information to a patient is the best way to change the behavior of the patient. And that's what we have implemented in our adherence platform at Ardex. And we have used a Six Sigma approach, saying we need to define the problem. Is it a problem of initiation, implementation, persistence? We identify the best measure, we analyze this information, and we provide the healthcare professional, the investigator in the case of studies, with all the tools, with all the tips and information to discuss, to focus the discussion with the patient to improve his medication adherence. And that has shown to be very successful. And we have to realize that the pharma model is changing. And maybe that one dose may not fit everybody in the future. The optimal dose of a population may not be the optimal dose of each individual. And that's where I think we are moving more and more towards precision medicine, personalized therapy, individualized treatment, patient-centered care, mHealth, eHealth, but also the payers. In the US, it's becoming more and more frequent for expensive therapy to pay for performance and to have more individualized treatment and treatment that are well taken will become critical in this new model for the pharma industry. And medication in adherence in that setting which will really become a vital sign to measure and manage from the trial process to the medical practice. So we are really in the age of patients empowerment, and I think that adherence-informed clinical trials will deliver better efficacy, lower variability, which will increase the power and decrease the sample size of trials in the future. It will deliver, and that's really important, better informed benefit risk and development decisions. And especially in a context where we are moving to adaptive clinical trials and more trials where decisions will be taken on smaller sample size, it will be key to know how the patients took the medication during the trials. We will have a faster proof to efficacy if we manage adherence, and then it will require validation in broader population. We will have less trial failure. And it's only if we work on that in the trials that we will be able to deliver in the medical practice more informative safety, more effective dosing regimen that will enable individualization and personalization of therapy, and it will facilitate a multidisciplinary approach. And that is really the key in the future, is that physicians will not manage adherence in the future in daily practice. But it's really a multidisciplinary approach where pharmacists, nurses will work together with physicians to optimize treatment, and adherence will be a vital sign in that picture. At Ardex, we are a global provider of adherence solutions, and we have developed an ecosystem where we have our own devices based on the MEMS technology, but we are opening the platform to any other devices of given quality and that are compatible, so that we can provide independently of the device, we can provide appropriate analytical and visualization tool to measure and manage adherence for healthcare providers to patients and for the pharmaceutical industry and the payers. With that, I would like to conclude by saying that we can have the most effective therapies available, but if we don't have appropriate adherence to medication, Did you lose it a long time ago?
No, it was just basically when you were talking on this slide.
Okay, and this is my last slide. So I just wanted to conclude, don't forget, drugs don't work in patients who do not take them, and it's key to measure that aspect of medication adherence in clinical trials and in medical practice. Thank you.
Thank you very much, and sorry for that last bit of problem with the microphone. Professor Ryans, that was a very interesting talk. For me, I've been involved with clinical trials for a long time, and you had mentioned I was in psychiatric studies, so in depression, and
Yep.
the adherence to study medication is just known that people just don't take it. You talked about your group creating the MEMS, and you showed us a slide. I think it was just the slide before this. If you could show us again, I'm curious to know exactly what that looks like. Is it something that's attached to the package? What is the
Yes.
technology that you use? Can you explain that a little bit more?
Yes, sure. So the idea is to put a chip in the package. So the original MEMS is a standard bottle with a closure that contains a chip, and we will detect systematically the opening and closure and time between opening and closure of the cap. So we have some algorithms to make sense. You know, if you open too fast, too slow, and so on, we have some algorithms to make sense of those data. And we have used it in, I would say, more than 1,000 patients pharmacokinetic studies, so PK studies. And we have about 97% accuracy between opening the cap and blood concentrations.
Okay, so that actually works much better than the regular self-reported or the diaries.
Yes.
Do you envision anything in the future? I don't know, maybe if there's going to be more. Let me back up. It's very important to have the adherence to the medication, but you also in clinical trials then do that measurement of the drug levels in the blood, like you said. Do you happen to know if there's going to be any future, I don't know, non-invasive ways to measure PK levels in blood or anything? Or are we always going to have to
Yes.
yeah.
No, it's clear that the measurement of blood concentration is moving fast, where we have dry sample blood, and multiple companies are developing that. And I think it will be first used in the trials to have more often blood concentration, but also if we go to a more individualized, personalized treatment in the future, patients will probably be able to measure themselves and determine what's their own therapeutic window to have their own dosing adjustment.
Okay, that's really interesting in the future, but that would still also go hand in hand with MEMS. For
Yes,
example, if
the
you go
idea is
into
to
that real
combine
personal,
both.
yeah, exactly. So that really personalized medication, the patients will go ahead and test out, figuring out what their right dose is, and then the MEMS is going to, in fact, help them continue the adherence.
Yes, because today the issue that we see is that if you develop one dose fits all, the dose has there is a tendency to select a dose higher than needed to cover the population and to cover all the patients who have suboptimal medication adherence. So so that means if you go with that dose on the market, and this is what is happening today, the most motivated patients and the most adherent patients are overdosed and they get side effect and they discontinue.
Right.
So
Okay.
in the future, we need to be much more precise, and the patient who is really a good adherer can go ahead with a lower dose.
Which, of course, is better for their health, but also for their pocketbook in a sense,
Exactly.
because they're not paying for medication that they don't need. Okay, very great. Lovely, lovely to speak with you. I don't see any more questions on the chat room. Give a few seconds to, if you have some questions, write them down. But I want to just say thank you very much, Professor Franz. This was a very interesting talk, and thank you for sharing your views on the adherence. And if any of you are interested in looking into the technology, you can go ahead and contact the ARDEX group. Additionally, if you're interested in learning more about this type of adherence management, you can contact and provide us with the feedback and leave information at the info@eccrt.com website. So we'd love to have your feedback. If there's anything else that you would have liked to have covered in a webinar such as this, please send us that information. Thank you very much. Goodbye.
Goodbye. Thank you.